Yes, sir. Actually, I missed this class. I was
Okay.
having.
Then we have to do today from here onwards, right? Okay, so let's start and let the other to join because they are late now. Fine. So xylem, let me give you a glimpse of last class. So where we ended with. So in last class, I told you that there are the complex tissues. So complex tissues are those tissues, permanent tissues, which are made up of more than one kind of the cell, right? So complex tissues are made up of more than one kind of the cell. Here, the xylem are made up of four different type of the cells. Vessels, they are dead. Tracheid, they are dead. Xylem fiber and xylem parenchyma. Xylem fiber are sclerenchyma only. We have already covered sclerenchyma in detail. Xylem parenchyma, they are the living cell. We have already covered the parenchyma because they are the simple permanent tissue, right? So, but vessels and tracheid, here we have mentioned over here that these are the tracheid, these napiform cells, these elongated cells, they are tracheid. They have the pits and they are the perforated cells, tube-like cells. They are called vessels, right? So their function is to take the water from the root to the top. Then it comes to the phloem. The function of phloem is to transport the food from anywhere, right? From the stem to the top, or sometime from the top to the, most of the time from the top to the root, right? So their function is to transport the food material. They are also made up of four different kind of the cells. That's why they are called permanent complex tissue. Seed cells, companion cell, xylem fiber, phloem fiber, and phloem parenchyma, right? So these are the four kind of the cell. Here again, the fiber and parenchyma, they are same in xylem and phloem, and they are the similar. Fiber are made up of sclerenchyma, and phloem are made up of parenchyma. Again, two different kind of cells are there: sheep cells and companion cells. Sheep cells are partially living. I have already told you that they are the only cells which have the nucleus, but they, again I'll explain it because today we are going to study the phloem, right? Sheep cells are 50% living, 50% dead, right? Sheep cells have the protoplasm, but they do not have nucleus. Again, if we talk about the companion cell, they have the protoplasm, right? And their function is they have the protoplasm and nucleus, and they are completely living cells, right? These small cells are companion cells, right? And they are the sheep cells, right? Sheep cells are controlled by companion cell only. Fine. So this was the composition of xylem and phloem. Remember? So this thing we covered in last class. Then we covered the xylem part as well. Fine. So xylem, I told you that xylem are made up of four kind of the cell: xylem parenchyma, xylem fiber, right? So that we have already discussed, and tracheid vessels, right? So I have told vessels, the xylem fiber, xylem parenchyma, and then I told you the arrangement of xylem, endark and exarch, right? So endark, the metaxylem are outside, means new xylem are outside, and old xylem are in center. In exarch, the old are in center and new are in outer side. So that is the arrangement of xylem that is found in different plants. That we did. Today we are going to do phloem. So we will start from the phloem onwards, right? Let's see how much we cover today. So look, today we are talking about the phloem. So again, the phloem is made up of four kind of the cell, right? So what is the basic function of phloem? The basic function of phloem to transport the food, right? And phloem are made up of phloem constituent. Constituent cell means those cell which make the phloem. So as I have told you, this is complex permanent tissue. Permanent tissue never divide, and complex, they are made up of more than one kind of the cell. So first phloem cell, that is the sieve cells, or we can say tube cells. Tube cell. So first cell is tube cell. Look, these are called sieve tubes, these cells, right? So sieve tubes, this is called sieve element. Try to understand. This is sieve element. Sieve element or sieve tube is same thing. Sieve tube. Let me write it down. So sieve tube or sieve element or sieve cell, they are the tube cells or sieve tube, or we can call them sieve element. Sieve element. Fine. So they are the, what is the peculiarity of them? Try to understand. They have the peripheral protoplasm. So what is in center? In center, there is this lumen. This lumen is made up of a large vacuole. Right? There is a vacuole, right? This is peripheral cytoplasm. And one more important thing, there is no nucleus, right? I'll dictate all the things. So there is no nucleus. This is the no nucleus. This is the only plant cell which is living and do not have nucleus. Like we have discussed in the last class, when I think this Martin, where is Martin? Martin, are you there? Yes, Martin is there. So Martin told me that I asked that which is the animal cell which do not have nucleus in humans. So he told me that is RBC. So yes, RBC is the human cell, that is the living cell and do not have nucleus. In the same way, in the plant, this is the tube cells or sieve tube. They are the living cells, but they do not have nucleus, right? And protoplasm is peripheral. So they are living, right? No nucleus, they are living, and peripheral protoplasm. Peripheral protoplasm. One more important thing, nucleus is called the brain of any cell, right? So nucleus is the thing, like brain, our brain control our body. In the same way, every nucleus control the functionality of the complete cell. If the nucleus is not present, who will control the, that is the very important point you need to understand over here, right? If this if nucleus is not there in sieve tube or sieve element, which thing will control the functionality of these tube cells? So basically their functionality is controlled by these cells. These are the companion cells. You can see companion cells. So companion cells, so the second component of the phloem is companion cell. Companion cell. So companion cells, they have the proper protoplasm, they have the proper nucleus, right? You can see. So they have prominent nucleus. Prominent nucleus is present, right? Protoplasm is present. Protoplasm. And they control the functionality of these, right? This one. So that's how they help the sieve tube element, right? Why we call them sieve tube? Because they are the tube-like structure, and at the end of every cell there is sieve plate. Sieve means perforated plate where pores are over there, right? Sieve which you use to like clean the floor and all that, right? You must have seen the sieve at your home. The same kind of the structure is found over there. That's why we call them sieve element or sieve tube or sieve cells, right? That is the reason behind it. Between the one more very important thing which I want to discuss with you, between these companion cell and sieve tube, there are some cytoplasmic interconnections. They are called plasmodesmata, right? So plasmodesmata are the pore-like structure. We have already discussed number of time what are the plasmodesmata. So there is pore-like interconnections between these two cells, which interconnect these both of the cells, right? So because of this plasmodesmata, the protoplasm of both of the cells remains interconnected. That's how the companion cell control the functionality of tube cell. Is that clear?
Yes, sir.
Okay. Now, what are the rest of the component? There are again three component, two component more, right? So the third component is phloem fiber. So phloem fiber, nothing but they give the strength to the phloem, right? These phloem fibers are called sclerenchyma. Sclerenchyma. One very important thing which I want to tell you, which I want to share with you, you won't find this thing in book. basically the jeans pant which you use, right, the linen which you use, they all are phloem fibers, right? So the coarse fiber of pant, like rough pant or trousers which you use, uh, if they are not made up of cotton, like they are not very thin, if they are a coarse fiber, all these coarse fiber, they are made up of basically phloem, right? So that's how the economically phloem fibers are used. So they are used to making the string, they are used to making the thread, right? Sclerenchyma cells. So phloems are very useful. And when the plant is alive, on that time, their function is to phloem function is to transport the food. And these sclerenchyma provide strength to the phloem column, right? Again, last but not the least, that is the fourth one, that is phloem parenchyma. We know phloem parenchyma. Phloem parenchyma is living cell. This is the same parenchyma which we have studied in detail in simple permanent tissue. Their function is they are living, and their function is storage of food. Clear? Is that clear? Phloem is clear or not? Tell me. Clear to all of you?
Yes, sir.
Okay.
Yes, sir.
So please now note it down, because start, put the heading. One more important thing that is left. That is the most important thing on this the phloem topic. You will find this question in your medical examination or some other MCQ examinations, right, where the MCQ question comes. So phloem are also known as the bast phloem fiber, right? So phloem fiber, like I have told you that phloem fiber are used in making jeans pant, making coarse pant, trousers, and all that, and some string, right, rope, and all that, right? So phloem fiber are also known as the bast, B-A-S-T. Remember, so if the bast term is written anywhere, it means they are talking about the phloem. Okay, that you need to remember. Don't forget it. You will find this question. You'll go through with. So write down some of the facts on this. So first heading will be phloem. Please start noting them. Phloem. It is a complex tissue. It is phloem is a complex tissue that transport. Phloem is a complex tissue that transport organic food, that transport organic food, organic food inside the body of plant, inside the body of plant. Full stop. They are also known as bast fiber. They are also known as bast fiber. Full stop. Next, it is made up of. It is made up of 4 different type of. It is made up of 4 different type of constituent cell. Constituent cell. Now you have to put subheading, right? Sieve tube first. Sieve tube or sieve element. Sieve tube or sieve element. They are elongated. They are elongated tubular. They are elongated tubular conducting tissue. They are elongated tubular conducting tissue. Full stop. Sieve tube are placed. Sieve tube are placed end to end sheep tube are placed end to end, and they may be end to end, and they may be transverse or oblique. They may be transverse or oblique. What does it mean? Let me explain you what does it mean. Basically, when I say that they may be transverse or oblique, so their end may be like this way. This is the one sheave element or sheave cells, whatever you understand. So, and there is a perforated plate at the end. After that, they are placed end to end. So after at the end, there will be another cell. But generally, they are oblique. So this part is oblique, right? So their end will be oblique. That's what I want to say. Next point. They have many small pore. They have many small pore or sieve or sieve pit. They have many small pore or sieve pit at the end of cells. At the end of cells, you may see these are the sieve pits, right? Next point. The cells have peripheral protoplasm. Cells have peripheral protoplasm without any nucleus. Cells have peripheral protoplasm without any nucleus. Full stop. Next point. Sieve tube take part in conduction of organic food. Sieve tube take part in conduction of organic food, right? Sieve tube clear? Now, this companion cell is more important than sieve tube, right? Guys, again, I want to tell you that today I am teaching you, but I don't know how long I'll be able to teach you because still the screen is blurred to me. Right? Still, my eyes are not working properly, right? So just I am trying to manage it, right? And let's see how long I can go. Fine. So don't find.
Okay.
Because it will take more 2 or 3 days. I hope it will take more 2 or 3 days to become perfectly clear. Then the next one is companion cell. So companion cell, I have already told you that their function is to control the functionality of sheep tube. Fine. Why I am emphasizing it, because you can get the question, draw the well label diagram and describe the structure of fluid. Or if they will ask you this question, it may be 3 marker or 4 marker question, right? Again, if they ask you that what are the companion cell, describe them, right? Then you have to write it down some point, right? So that will be 2 marker question, but definitely they can ask you from here, sheep tube, sheep element, or companion cell. Generally, this is the preferred question of teachers. Fine, then you have to write it down. And one more thing, whenever I give you the question, please try to draw the diagram, right? And try to write correctly or elaborate, right, in the right way. I have given a model answer as well, right? If you'll ask me, I'll write down an answer, right? So please, whenever you get the test on the Friday, do it honestly, scan it, send it to me. I have checked multiple times, you must have seen that I have checked multiple times, and I have find that now still you are lacking with writing skill. And if there will be any problem in your writing skill, I'll take one extra class for the writing skill. But please write correctly to get good marks. Next. So next is the companion cell. Companion cell, I have told you that they have the prominent nucleus and protoplasm is present. So please note down. Companion cells are narrow. Companion cells are narrow, elongated, and thin wall living cell. Thin walled living cells. Full stop. They lie on the side of sieve tube and closely associated with them through plasmodesmata. I have already told you what is the function of plasmodesmata. Right? What are the plasmodesmata? Next one. The cells have prominent nucleus and dense protoplasm. The cell have prominent nucleus and dense cytoplasm. Companion cell, that is very important point. What is their function? Companion cell help in maintaining pressure gradient, in sieve tube element, Clear?
Sir, could you just tell me what plasmodesmata is?
Yeah, look again. Let me tell you. Plasmodesmata. Suppose that there are two cells into two plant cells anywhere in plant cells, right? So if two plant cells are interconnected with each other, so between two plant cell there will be some pore or hole. So they are the connections. Like you must have seen, like there are suppose that there are two rooms in your home. There are two rooms, and both rooms are interconnected with a window, right? So the air can go here and there, but that is a small window. Through those window, air can pass. You can pass a small thing, pen and all that, a small object, but you cannot pass through them the table, chair, laptop, and all that, right? So number of the things, like the big things, cannot pass through, but they are interconnections. So those interconnections, those small window between two cells, are called plasmodesmata. Through the plasmodesmata, the material can be exchanged. Protoplasm can interconnect, right? Protoplasm can exchange water, mineral, some nutrient, and all that. But mitochondria, chloroplast, nucleus, these big objects cannot cross through that, right? So those interconnections between two plant cells are called plasmodesmata. Clear?
Yes, sir.
Martin, we have discussed already plasmodesmata, and one more thing I have already told you, that the interconnection between two animal cells are called, what we call them? Martin will tell me, Alia will tell me, and Yashir will also tell.
Yes, sir.
What we call, like the interconnection between two plant cells are called plasmodesmata. desmata. What we call the interconnection between two animal cells. Come on, guys. Don't disappoint me. Did we discuss it or not? That I also want to know. Did we?
I don't think we did.
No. We call them cellular interconnections. Simply note it down. Please note down. The junction between two cells. The junction between two cells are called. The junction between two cells are called cellular interconnection. Fine. Clear? Okay.
So like the interconnection between two plant cells is plasmodisplasm.
Plasmodisplasm. So the term is different, the function is same, right? And when I will teach you more animal anatomy, then I will tell you what are the junctions. But basically, junctions are plasmodispata are of single type. They are just plasmodispata. But cell junctions or cell interconnections, which are found between the two animal cells, there are some varieties, right? In the nervous system, I'll tell you there are chemical junctions, there are electrical junctions, right? So different type of junctions. But cellular interconnections are the term that we use in place of plasmodispata. Fine. Again, phloem
Sir,
fiber.
could you just repeat it?
Yeah.
The junction between two cells are called?
Two plant animal cells. The junction between two animal cells are called cell junctions. Cell junctions, right? Come to the phloem fiber. So phloem fiber I have discussed. They are the sclerenchyma, right? And they are dead, right? So please note it down. Phloem fiber. They are sclerenchyma made up of dead cells. They are also called bast fiber. They are also called bast fiber. Phloem fiber provide mechanical strength to phloem column. They are used as textile fiber. They are used as textile fiber. That you need to remember. These are the questions which are neat questions basically. What is the bast? Bast are basically textile fibers are the bast, right? What are they? They are basically phloem fiber made up of sclerenchyma. So that's how they ask the question, right? Neat questions are not difficult questions, but these questions need conceptual clarity. When you will have conceptual clarity, you will be able to answer those questions, right? Now come to the phloem parenchyma. So simple. Phloem parenchyma. They are living, elongated, ordinary parenchyma cell. Ordinary parenchyma cell having protoplasm, and connected with plasmodesmata. Plasmodesmata.
Sir,
Right?
could you repeat the second note, the second point?
Okay, fine. What I say? Okay, they have have protoplasm. They have protoplasm and connected with. They have protoplasm and connected with plasmodesmata with other parenchyma with other parenchyma cells. They are absent in very important question. They are absent in most of monocot plant. Means this phloem of monocot plant mostly missing the phloem parenchyma component. Means basically there will be only three components. Is the phloem clear or not? Tell me.
Yes, sir.
Great. Alia, clear?
Yes, sir.
Yasir? Great. Now come to the tissue system. Okay. Now come to tissue. Tissue system. So basically there are three tissue systems, right? So rather than do system name, tissue system are of, we have categorized them to study three categories, right? Ground tissue, vascular tissue, and dermal tissue. You know, derma, you understand, there is a term derma. Derma stands for skin, right? So those tissues which make outer skin of the plant, they are called dermal, right? So we call them dermal. So first category is that is the dermal. Fine. Dermal tissue cells, they are epidermal cells, stomata, and trichome. So epidermal cell like our skin. Stomata, like we have pores in skin. So same type pores are found in the plant for the transpiration. We do use the same pores for the transpiration, right? Trichome, like we have hair on our body. In the same type, plant stem have some these hair-like structure, hairy structure. They are found in our skin in humans. The same kind of the structures are found in the plant. We call them trichome. Clear? I'll tell you each and everything in detail. Right now, here I am just making you understand what exactly they are. Then come to the vascular tissue. So what are the vascular tissue? Vascular tissues are basically xylem and phloem. Their function is conduction of water and food. Xylem and phloem we have already. So trachea, vessel element, chief tube, and companion cell. It is written over here already, and we have discussed them in detail. Third, the last but not the least, the third one, that is the ground tissue, right? What are the ground tissue? Now, between the dermal tissues, means outer skin, and between the central part, that is central column, xylem and phloem, between them, there are some tissues which remains filled in the plant stem and the root as well, right? So we call them ground tissue, right? Basically, 70 or 80% of the ground tissues are made up of parenchyma. Parenchyma or filler tissue. Wherever in the plant there is a some like lacuna is there, right? Or some spaces are there, they'll be filled by these uh, parenchyma tissue. So ground tissues are parenchyma, collenchyma, and sclerenchyma. They make the ground. Clear? Now we discuss one by one. It is written parenchyma and sclerenchyma are also associated with the xylem and phloem, right? We know that. So now we will discuss one by one. Clear? First, please note down what are the tissue system. I'll dictate some. Tissue system. Tissue system is a tissue or tissue system is a tissue or group of tissue or group of tissues derived from meristem. Meristem is meristematic cell. Derived from meristem, which perform similar function, in plant body. Similar function in plant body, irrespective of their position. Right? Now we'll start with the dermal tissue or epidermal system, right? So first tissue system is epidermal tissue. Let me tell you first. Let me explain you what are the epidermal. I have already told you.
So I can show that
Previous
flow
slide.
chart again. Yeah.
Okay. Yeah, I'll tell you. Please note it down. Please note down this, that the tissue system, then ground tissue, vascular tissue, and dermal tissue. Don't write down this cells, the type of cells, right? Which comes under them. Done?
Yeah, done, sir.
Look. And they are called stomata. Stomata, you know. Stomata we will study again in chapter of transpiration in detail. What is their function, how they perform the function. Here just we are, for sake of understanding, we are just studying for what kind of tissue they are. So basically these are the stomata. Stomata are basically found in the lower surface of leaf, right? Their function is transpiration. Transpiration means evaporation of water, right? So water get evaporated from these pores, right? They are made up of guard cells. So these are guard cells, right? Stomata are these, right? These are the guard cells. So from these guard cells, water comes out. Fine. Then there are some unicellular hairs. These unicellular hairs, right? You can see some multicellular hair, right? So these unicellular and multicellular hair, right? They are basically these multicellular hairs. They are called trichomes. Unicellular hair, root hair to absorb water, right? Trichomes for protection, right? So these kind of the hairs are found. These all are, and one more thing, they are the epidermal cell, right? So epidermal cell, these cells are like this skin cells. They are epidermal. Epidermal cell, one very important thing additionally which I want to tell you. So suppose that this is the leaf. If you will see the leaf from the upper end, it will like upside, and this upside, this is upper end, and this lower end. So upper one is called adaxial. Remember this kind of term. Lower one is abaxial. So normally the lower end it have stomata. I have already told you for transpiration. If stomata will be on upper surface, more on upper surface, right? stomata are more in lower surface. Not like that upper end do not have, upper surface do not have stomata. They have, but very less. But lower surface are abundantly stomata. So basically the transpiration takes place from the lower end. Why in the lower end? Because if the stomata will be on the upper end, what will happen? The more transpiration will take place. More loss of the water will take place. So plant will face the scarcity of water. So to prevent the loss of water or extra loss of the water, the stomata are found more on the lower end or lower surface, right? Upper surface, it is or upper or lower, both kind of surface. They are made up of epidermal cells. So epidermal cells are these kind of elongated cells, and there are proper nucleus. Right? Yes. So upper surface of these epidermal cells, it is covered by waxy material. Wax, right? This is called cuticle, right? Cuticle. So this waxy layer, this waxy layer is called cuticle, and it is wax substance or fat. Is that clear? Done?
Yes, sir.
Please note down this whatever I have told you, right? Please draw this diagram what I have drawn right now. Cuticle, this waxy layer is also, this material is also known as the cutin. Cutin, remember it. Prevent loss of water. Let me know when you do. Done, guys? Put it down.
1 second, sir. Done, sir.
Fine. Martin, done?
Yes, sir.
Okay. So now you have to put the heading epidermal tissue system, right? After the class, you have to draw the diagram, right? These diagram, this stomata diagram and this hair diagram, right? Two things only, right? This one and this one. These two diagram you have to draw it. Please note it down. Put the heading epidermal tissue system. Epidermal tissue system is made up of epidermal tissue system is made up of two components. Two components. What are those two components? First component is epidermis, and the second component is called, what we call them, these here epidermal appendages. Epidermal appendages. These epidermal appendages, they are basically first trichome, epidermal cell. Epidermal, what is the epidermis? That I have already told you, right? I'll dictate, but try to understand. Trichome, and then the next thing which what I have told you, that this is the root hair. One more important thing, trichome is the characteristic of a stem. They are not found in the root, right? Root hair, you know, they are found in the root, but trichomes are the characteristic of a stem. If any object, plant object, it have trichome, it means it's a part of the stem. This is the fundamental of biology, right? So please note down first epidermis. Right? So first we will discuss about the epidermis. Is the epidermis is outermost. Epidermis is outermost protective. Epidermis is outermost protective layer of plant body. Epidermis is outermost protective layer of plant body. Full stop. It is usually single layered. That is important. It is usually single layered, but sometime can be multi-layered, but sometime can be multi-layered. Next, cells are compactly arranged. Cells are compactly arranged and living. Full stop. Cells contain large central vacuole and thin protoplasm layer right? Thin protoplasm layer. Next point. The outer wall of epidermis, is covered with cutin. The outer wall of epidermis is covered with Cutin, full stop. It is a waxy substance which prevent loss of water. Right? And next one. So next thing that in epidermis, the part of epidermis, stomata are the part of it. I think the stomata, you must be aware with the stomata. So they are part of epidermis. Fine. This diagram of stomata is important for your school examination. In school 30, they may ask you to draw the well-labeled diagram, right? If the diagram-based question comes at your school, definitely they'll ask, right? This is very important, right? So diagram is important. Look, basically the stomata is made up of these subsidiary cells. These yellow color cells, they are called subsidiary cells, right? And then there are two kidney type cells, right? These kidney kind of the cells, they are called guard cells. So there are two guard cells and surrounded by subsidiaries. Fine. These guard cells, they have thick cell wall inner side, right? And thin cell wall outer side, right? So, and the space between them, pore between them is called stoma, right? Stoma is that is the pore. This pore is meant for exchange of gases, So what kind of the gases? When we talk about the exchange of the gases, it means I am talking about the entry of carbon dioxide. So carbon dioxide will come in and the oxygen will go out. So that is the exchange of the gases. Then transpiration, removal of the water, transpiration. So that is the function of stoma. Then there are chloroplast is found, that is important. Chloroplast is also found in this guard kind of the cell. They have the nucleus, right? And the subsidiary cells are surround them. Fine. So this is the stomata. So after the class, you have to draw this diagram. Clear? Fine.
Yes, sir.
Okay. So what is the next? So, please write down few points for the stomata. The epidermis usually bear the number of minute pore, called stomata, full stop. Stomata are absent in layer of root. Stomata are absent in layer of root. Full stop. They are made up of two specialized guard cells. They are made up of two specialized guard cells surrounded by guard cell surrounded by subsidiary cell Full stop. They are meant for exchange of gases They are meant for exchange of gases and transpiration of water. They are meant for exchange of gases and transpiration of water. So the second subheading will be epidermal appendages. Epidermal appendages. In epidermal appendages, so there are first there are trichomes, right? So that please load it down. A. So A are trichome. Trichome, I have already explained that they are found in the surface of the stem, right? So please note out. They are unicellular. They are unicellular or multicellular. They are unicellular or multicellular outgrowth of hair. Full stop. They are strictly epidermal in origin. Epidermal in origin means they will come out from the epidermal cell only. They are strictly epidermal in origin. Their function is protection. Their function is protection. Right? Come to the second one. Root hair. Next one is root hair. Root hair that we have also discussed. They cannot be multicellular. They are unicellular tubular structure. They are unicellular tubular structure found in the root hair. Found in root hair zone of root. Next one. So basically, root hair are like this one. This is epidermis, right? This is like epidermis, and out of any of the cells from the epidermis, this hair-like structure will come, and they will make the root hair. Right? They are root hair, and then root hair, their nucleus will be like this one. They will have nucleus. This is epidermis, and this is root hair. Right. So please note down. Nucleus is found at apical part. Nucleus is found at apical part. Full stop. They are epidermal in origin, means do not originate from inside. They are epidermal in origin. Is that clear to all of you? Epidermal tissue system.
Yes, sir.
Great. Martin and Yasir. Alia,
Yes.
all clear?
Yes, sir.
Was you absent for a few minutes?
Yes, sir.
So is there any doubt or confusion?
No, sir. Just for 2 3 minutes, actually, sir.
Okay, great. So so this was the epidermal. Now come to the ground tissues. We won't be able to complete this system today, right? But ground tissue system, look. Try to understand this central part is made up of this central part is made up of vascular system, right? This central this part. This is vascular system, right? Epidermal tissue system we understood, right? So at the center there will be some vascular system. What is the rest of the part? The rest of the part is called ground tissue system. So epidermis is not the part of ground tissue system, right? That is a part of epidermal system. From cortex onward. So cortex is a part of ground tissue. Medullary rays, they are these rays like structure, these green structure. They are part of ground tissue. Pith is part of ground tissue. Pericycle is part of ground, right? So these all ground part are called ground. Fine. So we include them in ground tissue system. But this was the part of the ground tissue system. So let me tell you, ground tissue system of stem. But when we talk about the ground tissue system of the leaf, this is the ground tissue system of the leaf. So ground tissue system of leaf. Please focus over here. I will ask all these questions which I am telling you today, which I am teaching you today. I will ask all these questions when I will teach you photosynthesis part, because in photosynthesis the structure of the leaf will be important, and there I will ask you all these questions. So do not forget to ask, right? Do not forget to remember them, right? I'll definitely ask you. So again, waxy cuticle, that is the epidermal part, right? So this is upper epidermis. So this is epidermal system or dermal system. Right. This is palisade mesophyll. Palisade mesophyll. Yes. Palisade mesophyll are basically uh, the upper part of the leaf. So these cells are compactly arranged, right? You can see there are these green dots are basically they are the chloroplast. Chloroplast, right? So these are the chloroplast, and this is the palisade mesophyll, and there are spongy mesophyll. Spongy mesophyll, they are the also cells, so there are a space between them, right? Air spaces are there, right? So and they are not densely covered, and there are air spaces. This is the lower epidermis, waxy cuticle, right? Again, there are the guard cells. So you know all these are the part of again epidermis. Right? Out of them, there are only these two cells. They are the ground tissue, right? Okay, clear?
Yes, sir.
Okay. So fine. Please note down some ground tissue. Put the heading ground tissue. The ground tissue system is formed from ground meristem. Ground meristem. Right. Ground meristem. Fine. Next one. They are made up of simple parenchyma. They are made up of simple parenchyma. Collenchyma. Sorry, not collenchyma. Parenchyma. Collenchyma and sclerenchyma, right? And sclerenchyma. Fine. Next one. The ground tissue system. Of leaf. The ground tissue system of leaf is called mesophyll. The ground tissue system of leaf is called mesophyll. Full stop. Mesophyll is made up of two type of photosynthetic. Mesophyll is made up of two type of photosynthetic cell. First, palisade mesophyll. Second, spongy mesophyll. Right? They are meant for add on one more point. They are meant for photosynthesis. Next heading, ground tissue system. Ground tissue system of stem. It is made up of following constituent. It is made up of following constituent. First, hypodermis. Second, cortex. Third, endodermis. Fourth, pericycle. Fifth, and sixth, medullary rays. Okay, right? Note it down, and we'll do it in next class, right? And you will make me recall that we did till this part.